Hello friends, welcome to my YouTube channel New Biologist. I'm starting with a new topic called the manipulation of purified DNA, or else in other words we can say that what are the enzymes involved in rDNA technology. So I have planned to take this whole topic into four parts. So let's start with the first part. Here we will see the range of manipulating enzymes. What are they? So when we are looking for the enzymes, we can classify broadly in four classes. One is like nucleases, polymerases, modifying enzymes, and last is ligase. So what are each and their role? Nucleases that cut the or shortens or the degrade the nucleic acid molecules. So we are going to look the DNAs, then the restriction enzymes. Then the next point is like a polymerases that makes the copy of molecule, modifying enzymes that remove or add the chemical group, and last there is a ligase that going to join the two different molecules together. So let's talk about the first nucleases. So there is a two type. One is called the exonuclease, another one is an endonuclease. So what are the characteristics of both? So exonuclease that removes the nucleotide at one at a time from their end. So exo at the end, endo it's going to be in between. So here the endonuclease that can break the phosphodiester bond between a molecule within itself. So let's see one by one. Here the picturization is going to see show you us very well. So exonuclease you can say so it's going to cleave a bond between like you can say here the one of the nucleotide is having phosphodiester bond between two adjacent is there and one hydrogen bond is there. So exonuclease is going to cut both phosphodiester bond as well as the hydrogen bond. From the both the end cutting will be start as the time will be permitted more for the activity the exonuclease is going to degrade the whole DNA in given time. In case of an endonuclease, it is going to cut the phosphodiester bond between the two adjacent nucleotides. So if the time permitted very high, here we can see with the red arrow somewhere it is cut one to second and third. So many smaller fragments can be established. There are the two main important you can say the exonucleases are there. One is a BAL3 one, another is one uh, E. coli exonuclease 3. What are their activity? BAL3 one, which is well known for their exonuclease activity from their end. It is going to cut the DNA from their end. While exonuclease 3 having the characteristic is that it is going to cut one by one nucleotide from both the 3 prime end. Yes, here you can see the 3 prime end is there and another is going to remain hanging. So it's became like a single stranded DNA. Similar to it, one of the well-known is like S1 endonuclease that only give the single-stranded DNA. Earlier, what we have seen, both were working on the double-stranded DNA. Here, S1 nuclease is working on single-stranded DNA. So we can see here it is that first part. Single-stranded DNA is there. It's cut into various. And if there is a double-stranded DNA, but it having the nick region, so this nick can be cleaved off or the cut by the S1 endonuclease enzyme. Another example is a DNase1, which has been isolated from the cow pancreas, and it can cut both single-stranded as well as double-stranded DNA. And as we are going to keep the single-stranded DNA or double-stranded DNA for prolonged time in the conditions with the DNA1, it's going to generate some mononucleotides, or you can say the very short oligonucleotide can be formed. Here we are going to learn the very specific class that is called the restriction endonuclease, which cuts the DNA of the double-stranded DNA in very specific pattern. So there is a very well-known phenomena that is if the restriction enzyme is there, why it's not going to kill itself or the kill their own DNA. So here we are going to learn the one basic phenomena called the host control mechanism, how it come to know that earlier it has been observed that the bacteria when infected by the phage, if the bacterium having the some restriction enzyme, so they are going to cut such a phage inserted DNA and there is a, you can say the immunity is gained by it. But it is not going to affect their own DNA. So why it is not so? So bacterial DNA is not get cleaved because there is a some methylation is taking place on the DNA which masks a restriction enzyme site to be get absorbed by this restriction endonuclease, and they always remain survive. Okay. So there are the what are the types of the restriction enzymes are there and how the cutting is taking place. There are the two different kind of a cuttings end generated. One is a blunt end, another is a sticky end. So first we will look for the blunt end. Okay, so it is been known like a PU, a PVU2 and the ALU1. They generate the blunt, or you can, another terminology is called the flushed end is generated. So here we can say that the production of blunt end, there's some nucleotide is there. So when it's going to look their specific restriction enzyme site, it is going to cut into two halves. So blunt end is going to be get in two halves is generated. Another is called the end is generated sticky end. Also, people are calling it as a cohesive end. So here the cutting is happened in a way that some of the strands are remain hanging from one to another. So here we in picture we can see like eqr1 has having their own one restriction enzyme site is there. When it is cutting, some overhanging regions are generated, which is considered as sticky ends. So there are the when we are looking for more in a detail, we can see there are the three types of restriction enzymes out there. One and two, 1, 2, and 3 is there. So type 1 and 3 having certain limited role because there are the sometimes the restriction enzyme sites are there, but they are not acting on their own place, or but they may cut far away from their own region of the restriction site, or they are non-specific. But yes, type 2 is very specific, which having the restriction enzyme cutting site, and over there only they are going to cut. So some very well-known, you can say the restriction enzymes are there. Among them, certain are generating blunt end, another is the sticky end. But here I would like to highlight four major. This has been asked in the many a time in exams for their, uh, you can say the restriction enzyme sites are there. So EcoR1, please, friend, keep in your mind this kind of a restriction enzyme site. That is EcoR1 is a GAATTC. I repeat, GAATTC. Palm H1 GGATCC, GGATCC. In 3 is AAGCTT, and last one is a HAE3, which having a very there is a 4 nucleotide only, GGCC. So I will tell you people that you may write 2 to 3 times or try to recall or remember by your own the restriction enzyme sites. And in my next video, I will take you for the polymerases, that is in a part 2. Okay, so this is the one of the video for the modifying enzymes. If you have liked it, then you may subscribe, share with your friends, colleagues, or any students, those who are looking to prepare for the exams as well as to learn into such kind of topics. And if you wish to give any comments, please put into the comment box. Thank you.